Hello everyone. I am Gagandeep from Cybersecurity branch of Sri Venkateshwara College of Engineering. Today we are discussing about DNA in vaccines and diagnosis. Let's take the rabies as example and let's see how DNA vaccines works for it. So first of all, what is rabies? It is a zoonotic disease which nearly causes 59,000 human deaths globally each year. So it primarily um, transmits through animal bites and causes both wild and domestic animals. So the need of more efficient vaccination strategies is required. Let's see the benefits of DNA vaccines. DNA vaccines offers enhanced stability and they are cost efficient and they are safe as they do not use live pathogens. DNA encoding rabies. The DNA vaccines contain circular piece of DNA which surrounds the rabies virus glycoprotein, an essential antigen for immune response activation. This design allows the host cells to produce the antigen promoting an immune reaction against the virus. Intramuscular injection. So the vaccine are injected through the muscle tissue of the host body for effective delivery of genetic material directly into the host tissues for optimal immune response. Antigen production. Once the vaccine is inside the host cells, the DNA undergoes the process of transcription, which converts it into mRNA, which is translated into rabies virus proteins. These proteins are displayed on the surface of the cells, crucial for mounting an immune attack. Immune response activation. The reaction of virus antigens triggers an immune response, activating both humoral and cellular immunity. This multifaceted immune activation is essential for neutralizing the rabies virus effectively. So, this is about the DNA vaccines. Let's discuss how RNA vaccines works by taking the COVID-19 as the example. Let's take the overview of mRNA vaccines. mRNA vaccines utilize messenger RNA strands to instruct cells to produce proteins resembling virus components. This approach circumvents traditional vaccine methods that often use weakened or inactive pathogens. Mechanism of action. mRNA vaccines function by introducing and synthetic mRNA into the body, promoting cells to produce viral proteins that active the immune system. This method enhances both immediate and long lasting protective responses. So, the RNA vaccines can be used for both immediate and long lasting protective responses. mRNA selection. The genetic sequence encoding the spike protein of SARS-CoV-2 is identified and selected, serving as the blueprint for mRNA synthesis. This targeted approach ensures a robust immune recognition and response against the virus. Next, mRNA formulation. Once synthesized, the mRNA is encapsulated in lipid nanoparticles, that is, wrapped in lipid nanoparticles, which protect it from degradation and facilitate its delivery into human cells, enhancing vaccine efficiency post administration. Vaccination process. It is same as the DNA vaccines. mRNA vaccine is administered through an intramuscular injection, commonly into the upper arm. This method allows the lipid nanoparticles to efficiently deliver the mRNA to nearby cells for uptake. The uptake process after injection, cells surrounding the vaccination site observe the lipid nanoparticle mRNA complexes. The mRNA then enters the cell, serving as the template for synthesizing the viral protein. And then the virus goes to the neutralization and the virus is end. So this is how RNA vaccines work. So now let's discuss about the comparative analysis of DNA and RNA vaccines. DNA versus RNA vaccines. DNA vaccines introduce genetic material that encodes antigens directly into host cells, triggering an immune response. So, what DNA vaccines do is they send the antigens directly into the host cells, which triggers the immune system and goes neutralization, and the virus gets killed. But while in the RNA vaccines, it utilizes the messenger RNA to instruct cells to provide viral proteins. Both approaches stimulate the immune system but differ in their mechanisms of delivery and production. Stability and storage requirements. DNA vaccines maintain stability at room temperature. They can be stored at room temperature, eliminating cold chain logistics. But RNA vaccines require refrigeration, making their distribution and storage more challenging, especially in remote areas. Production scalability. The production of DNA vaccines leverages recombinant DNA technology, offering cost effective scalability, while RNA vaccine production can be rapid but has complexities in formulation. Both methods are adaptable to meet urgent global health needs. Safety profiles. DNA vaccines pose minimal risk as they do not use live viruses or live pathogens, thereby reducing vaccine associated adverse effects. RNA vaccines also exhibit strong safety profile but may cause mild short-term side effects like fever and fatigue, reflecting immune activation. Regulatory considerations. Approval for DNA vaccines requires comprehensive data on safety and efficacy, which can be extensive. While RNA vaccines have also faced scrutiny but benefited from rapid development pathways due to urgent needs as during the COVID pandemic. So this is about the DNA versus RNA. Now let's see the future directions in vaccination. Innovations in vaccination technologies. Recently, the innovation went so far that they are doing the needle-free vaccine delivery systems and the use of nanoparticles for improved efficacy. For instance, lipid nanoparticles enhance mRNA delivery, enabling broader applications and better immune responses. Potential applications beyond rabies and COVID-19. So now in this video we have discussed about rabies and COVID-19, but there are other diseases such as influenza, Zika and HIV. These flexibilities allows for rapid response to emerging pathogens, potentially revolutionizing disease prevention. Addressing vaccine hesitancy. So, however, they can find new vaccines, but people do not believe in it. So, for clarifying their doubt, vaccine hesitancy remains a critical barrier to achieving herd immunity. Public education campaigns, transparent communication about vaccine safety and efficacy, and engagement with community leaders can help build a trust in vaccination. Importance of continued research and development. Ongoing research is vital for the evolution of vaccine technology, ensuring preparedness for future outbreaks. Investment in R&D can lead to improved formulations, safety profiles, and delivery mechanisms essential for global health security. So, thank you everyone. This is it for today. Please like, share, and subscribe.